The overall goal of this procedure is to present an optimized experimental approach to isolate embryonic tissues from the quail and chicken embryos. With this technique, we obtain pure embryonic tissues that can be used in gene expression studies and functional assays.
Tissues from quail and chicken embryos are isolated by microsurgery and subject to in vitro enzymatic digestion while preserving its biological properties.
After isolation, the three-dimensional preserved tissues can be used for high-resolution topographical gene expression patterns analysis. This approach is particularly useful for detailing in situ gene expression in embryonic territories otherwise inaccessible by conventional methods. In addition, transcriptome analysis approaches can also be applied in isolated tissues without requiring genetic markers, while providing a tissue-specific high-throughput omic analysis.
Alternatively, the isolated tissues from both species can be associated in an in vitro organotypic assay for 48 hours. This approach allows to study tissues interactions since quail and chicken cells can be discriminated by distinct nuclear features and molecular markers.
The capacity of the culture tissues to form organs can be further tested by in ovo assay. This method is, therefore, a useful tool for studying complex tissue interactions in developmental processes with highly dynamic spatial modifications and can also help to reveal the potential of specific embryonic territories to the formation of organs.
As an example, herein is described the formation of Quail-Chicken Chimeric thymus from isolated embryonic tissues. First, the endoderm of the third and fourth pharyngeal pouches, the prospective thymic rudiment is isolated from the pharyngeal region of quail embryos with three